Hi, my name is Harshita, and my project is on understanding the evolutionary development of radioresistance in the Arabidopsis thaliana. Over the years, there have been many nuclear accidents, such as Chernobyl and Fukushima, that release radioactive contaminants which can be harmful to many organisms, including humans. In fact, nearly one-third of the world's reported cases of acute radiation sickness have been due to Chernobyl. However, not all organisms are affected in the same way. For example, plant abundance increased in the years following Chernobyl due to the positive impacts of land abandonment and human evacuation, which has provoked many studies focused on supporting and opposing the biomolecular evidence of adaptation. Since ionizing radiation is a primordial stressor, humans likely already have some level of resistance to it. However, due to their immobility, plants have evolved the capacity to better and more frequently adapt to their local conditions. Some molecular mechanisms for adaptation arise from how ionizing radiation damages DNA. For example, damage caused by double-stranded breaks can be repaired by homologous recombination, which in turn contributes to genome instability and variation, serving as the foundation of adaptation. The radiation responsive paralogs, RAD-A, REC-A, and RAD51, promote this repair process. However, the significance of studies confirming lower recombination frequency during later years at Chernobyl is often debated because while it could be evidence for successful adaptation, this outcome can also be due to naturally lower radiation levels with the passing of time. Irradiation has also led to radiolysis, which is the dissociation of molecules resulting in the production of free radicals or reactive oxygen species. ROS harm key molecular pathways by disintegrating target proteins and increasing oxidative stress. Key antioxidant promoting genes, such as the CAT and FSD3, which are involved in interfering with the free radicals functioning, have shown to be upregulated in exposed plants. For my methodology, I selected candidate genes that had functions centered on these irradiation molecular pathways. This study will target three species which have previously been involved with studying the effects of irradiation due to their small genome size. Debates surrounding the A. thaliana have argued its relative levels of radiosensitivity versus radioresistance, with many conforming to its status as a model organism and abundance in hostile environments. Similarly, the B. rapa species possess economically beneficial traits in their small genome, and their high improvements with herbicide resistance may indicate mechanisms for adaptation to chemical stress and irradiation as well. When exposed to increasing UVB radiation levels, studies have shown the flowering and growth of the B. rapa to decrease, however, total seed production to increase, indicating that the species' fitness may be unaffected, an implication of adaptation. In addition, the B. oleracea contains a similar structural genome to the other two, but has expressed accumulation of flavonols, a class of flavonoids, with UVB radiation as well. These flavonoids are powerful antioxidant agents that effectively reverse the effects of oxidative stress discussed before. For this study, I sought to determine the presence or absence of positive selection, which would in turn indicate irradiation adaptation between branches of orthogroup gene trees and the three species studies, the A. thaliana, B. rapa, and B. oleracea. These were the 12 candidate genes selected from thorough analysis of background literature as well as database sites, all corresponding with functions related to irradiation resistance pathways. For instance, the A2G20620 gene encodes for the CAT3, an antioxidant catalyst discussed before. I was able to determine 7 respective orthogroups for 8 of the genes, with 2 genes being in the same orthogroup. A few steps involving data processing were run. First, I downloaded protein-coded FASTA genomes from Plant Ensemble, a sample of which is showed for the A. thaliana below. In addition, I accessed the OrthoFinder program through the iTerm2 software terminal. Then I used specified code to determine orthogroups and gene trees for the candidate genes listed before. These gene group text files were then visualized into gene trees using ICTree and ET Toolkit as shown on your right. Then I used separate nucleotide sequences to produce CDS files required for the alignment run on MAXI for each orthogroup. The alignment concluded how similar each brand sequence is to the others in the orthogroup and provided insight into specific genetic positions, such as mutations of evolutionary adaptation. The alignment also provided the next analysis step, PAML, with properly structured data necessary to perform a series of statistical analyses used to infer evidence of selection. Using this PAML program, likelihood ratio tests were run on 2 chosen branch-side models. The BSA1 was the null model, while the BSA was the alternative representing positive selection. The test determined significance of model fit for each branch within each orthogroup using this PAML code. The home correction was then applied on the p-values from previous PAML analyses in order to account for the error rate compounded from multiple analyses using this specified formula. Following the home correction on the orthogroup 13,135, which contained 4 total genes, the last branch satisfied the condition, concluding rejection of the null and selection of the alternative model for positive selection on the corresponding A thaliana gene. As my analyses further, the LRT will be run on all 7 orthogroups. The positive selection in the A22G23050.2 gene is significant because it encodes for the NPY4 protein, which is involved in the ubiquitone protein ligase complex responsible for the addition of ubiquitone groups onto molecules. Ubiquitone groups have several functions, such as being a main regulator of protein stability, stress responsive, and involved in DNA damage repair as well, which could indicate its role in adaptation mechanisms to radiation discussed before. In addition, this biomolecule has been proven as a possible therapeutic treatment for multiple myeloma. The evolutionary context helps us understand past adaptation as well as provide insight into future adaptation, such as what genetic or cellular level changes are required to evolve radioresistance. The results of this project can be applied to better gene therapy options for human disorders, such as ARS and multiple myeloma, through the use of orthologs to these candidate genes. In addition, the genes determined to show positive selection can help harvest crops that are better adapted to otherwise harmful conditions through GMOs. Similar to agriculture, these genes can also help plants withstand the unfamiliar environments during long manned spaceflights and survive, paving the way for further explorations. Thank you for listening.